Thank you so much to the organizers for inviting me. I'm going to tell you about gene therapy for Huntington's disease. You might have seen this in the news in late September last year. It made the top story at News at 1, BBC News at 1, 6 and 10, and then was all over the world. So Huntington's disease is a rare disease. It's a devastating rare disease, and it affects people in the prime of life. The mean age of onset is 40. It's a chronic disease that's been called a combination of ALS, schizophrenia, and Alzheimer's and Parkinson's because it affects all parts of the body and brain in similar ways to those diseases, but in one disease. So this is a lady, age 38. This is her 13 years later with end-stage Huntington's disease. It's monogenic. It's a rare disease. And we currently have no treatments. And it's caused by a repeat expansion in the first exon of the Huntingtin gene encoding a toxic protein mutant Huntingtin. And so this is from a review from a few years ago. I have some more recent reviews, but I use this one because actually the figure's relatively simple. And I'm actually going to talk about gene therapy using microRNAs, although there's a number of other approaches in development. And in contrast to what Simon was presenting in his beautiful talk, which is gene replacement therapy. Most of the examples Simon gave were for kids, where you replace a missing gene. And so that's gene replacement therapy. Doing and targeting toxic gain-of-function diseases, which tend to be adult onset, has a whole level of challenge in a different way, because you're basically trying to lower the level of a toxic disease-causing protein. So I'm going to tell you about AMT-130, and this is a one-time administration. It's an AAV5-delivered episomal DNA. It produces a Huntington-specific microRNA. It silences using RNAi, and it targets highly toxic exon 1 fragment, and it targets the full-length mutant Huntingtin fragment. And so there was about 15 years of preclinical data, and I'm just going to show you a couple of the preclinical papers. This is showing widespread microRNA Huntingtin transgene distribution in non-human primates after intrastriatal injection, and that's direct injection into the caudate and the putamen. And you can see here in the caudate and putamen, there are areas really badly affected early in Huntington's disease, very good transgene levels. And this is, I think, a really important study. This was in transgenic mini pigs because we needed to go to large brains because it's pretty easy to cure a mouse of Huntington's disease, but it's very difficult to translate that to an organ the size of the human brain. So we use mini pigs, which actually have quite large brains despite their name. And this was a study in transgenic Huntingtin mini pigs who get sick at 10 years of age. So it's still age-related in the pig. So this is a 6 months and 12 months. These are all the brain regions. This is mutant Huntingtin protein. And intrastriatal interparenchymal surgery in the pigs showed after 6 months very good lowering in the deep structures, which is what we want, and reasonable levels of lowering in the cortex. And it spreads from the striatum through retrograde transduction to the cortex. And so the path to the clinic, multiple preclinical models were used, mice, pigs, and non-human primates. The molecular mechanisms and the targets were fully validated from the preclinical studies in mice, particularly the exon 1 transcript. We had clear evidence of target engagement in pigs. And we tested the neurosurgical procedure in the pigs because this has been a novel neurosurgical procedure that's never been used before. 6 intracerebral catheters directly injected into the deep structures of the brain to get the gene therapy where you need it. And so the pigs were used to test the gene therapy protocol. And the PK/PD modeling gave very clear indications of dose response and also tissue pharmacodynamics. And the toxicology identified a very wide safety margin, so we went to humans. And so this is the surgery, and I had a recent meeting with the MHRA, actually, and they said they'd had no drugs ever had gone to them with gene therapy that involved deep structure, intraparenchymal brain surgery. So it was a new for them. And the protocol was developed by a very talented neurosurgeon, Brad Elder, in the US. And one catheter into the caudate, 2 catheters into the putamen bilaterally. And the surgery takes between, the quickest is 12 hours, and the longest has been 23 hours. And all the surgeries in the UK, we wanted to do them all in one place. Liam Gray went over to the US to train, and Liam did all the surgeries in the UK only really because there was only 2 other neurosurgeons who were able to do it. And it takes up an NHS MRI-guided operating suite for a whole day. So in the current NHS climate, it's challenging to get centres that can do the surgery. So this is actually a video with an MRI-guided enhancement. It's got gadolinium enhancement. This has been speeded up about 1,000 times because this actually takes several hours, because you have to inject through convection-enhanced delivery using the ClearPoint SmartFlow, and you need to do it very, very slowly. So this is speeded up, but actually, once the catheters are in, the neurosurgeons tell me that it's a very boring surgery because you have to sit there and infuse very slowly. So this is the surgery. And so I'm just going to, before I go to the study results, I'm just going to tell you about the cohorts. We had 2 cohorts, one in the US and one in Europe. And we treated people with early symptomatic Huntington's disease, and they had to have a certain size of their caudate and putamen to be safe for the surgery. As the disease progresses, you begin to lose more of your caudate and putamen volume, and by the time you're getting to advanced disease, you've really just got a sliver of it left. And so the study I'm going to show you is the cohort 1 and 2 combined, and the database lock was the 15th of August last year. And the primary analysis is those 2 study sets. So, and this has been mentioned in the first really interesting discussion. When you work with rare diseases, particularly for gene therapies, it is difficult to have a sham-controlled, 3-year randomized controlled trial with a placebo group. Because if you were a person with Huntington's disease, and I think morally, I think it's similar for many diseases, would you want to sign up to have your brain drilled into for maybe 20 hours with 20 hours of general anesthetic? Because it's a sham surgery, you have to have the same length of general anesthetic. It's just a burr hole, and then just be followed up for 3 years, having had a placebo. And then you may not be eligible for any other studies after that because it's a progressive disease. So the FDA insisted on a sham, but just for 1 year. And in retrospect, that sham hasn't proved that useful because it's too short for a gene therapy for a disease like Huntington's disease. But in the UK and Europe, it was open label. So because of that, we have to have an external comparator group. So for gene therapy studies, in rare diseases, it is not uncommon to have an external comparator group. And in the Huntington's field, we're lucky to have a very large registry of subjects with over 20,000 people worldwide, and that's what we used as the external comparator group. And we used a method called propensity score matching to actually match the external comparator group to the individuals. Now, I'm going to give you a very simple explanation of propensity score matching. So any of you who are experts in the statistics of propensity score matching and weighting, please I apologize for this simple explanation. So in clinical trials, we want to compare 2 groups: people who get the drug and people who don't get the drug. And in a traditional RCT, you have a placebo group, and that's the group that you compare to. But as I've mentioned, randomization or a long placebo is sometimes not possible. And that's where PSM comes in. And it's a method where you match and you mimic randomization by making the two groups as similar as possible. And in this case, we used the Enroll-HD dataset. We had many discussions with regulators. They were very clear we could not use a historical dataset. And we have some very good historical datasets, but it had to be a concurrent, living, ongoing study for the comparator group. And so this is an ongoing study, the Enroll Registry Database. So we use that as the external comparator group. And as I mentioned, it's an approved method for rare diseases. The FDA and EMA have approved a number of therapies with PSM or PSW matching for rare diseases. So we did a pre-specified pivotal analysis. We had 17 patients in the high dose, 12 in the low dose, and the primary was the change at baseline in a number of key clinical measures of progression. This is the baseline demographics. This is the high-dose group, the N of 17, which we used as the primary group. This is the comparator group, and they are matched for exactly the same characteristics, and there was no difference apart from most of the high-dose group were in the US, and more of the PSM-matched group were from Europe and the UK. So this was the primary endpoint that got everyone quite excited. So we have had no treatments for Huntington's. Every clinical trial so far has been negative for lots of different reasons. And so I've worked in the field for 30 years, and I was beginning to give up hope. But I saw this data at the beginning of September, and I thought, wow, I think this drug is doing something. So this is the high-dose group. It's only 12 people, but they are 12 people that are very typical of the disease. And this is the matched control group. And so even with just 12 people, it's statistically significant. And there was a 75% slowing of this measure, which is the United Huntington's Rating Scale Composite, which is function, if you're doing your job, how you're managing your daily life, thinking tests, and a motor neurological examination. So they're all key things that we measure. These are the actual plots. This is the external comparator group, and this is the active group. And you can see here, and actually lots of people have asked me in the spaghetti plots, is it driven by an outlier? But no, they're all really quite uniform. And so you can see very clearly the difference in the CUHDRS. Total functional capacity is a functional endpoint, whether you're in your job, can you manage your job, can you manage your finances? It's resistant to placebo effects. And here you can see the N of 12, a 60% slowing of a decline in total functional capacity. And again, even with tiny numbers of treated, it's statistically significant. And again, this is the spaghetti plot. This is what we would expect over 3 years in the external comparator. This is the real data. This is the active group. And you can see here at 24 months, they're beginning to improve. And 2 people have gone back to work. Now, 2 isn't a small number. It's 12 in total. But I have been looking at, I'm a physician scientist. I've been looking after patients for 30 years. I've never seen anyone give up work and then improve to go back to their original job with Huntington's disease. So this is, they're beginning to get an improvement. These are some of the other endpoints that were mentioned. This is a cognitive endpoint. It's a thinking task, symbol digit modality. And you can see here that there is a 88% slowing of a deterioration in the symbol digit. It's a psychomotor cognitive task. This is a Stroop word reading task, and you can see here this was reduced progression by 113%. And here, this is the total motor score, which is your neurological examination, and it reduced progression by 59%. And then one of the measures we use in neurodegenerative brain diseases is CSF neurofilament. It's produced by dysfunctioning, dying neurons, but also dysfunctioning neurons is a key area that produce the neurofilament. It's not just dying neurons, it's also dying and dysfunctioning axonal function. So CSF neurofilament is well recognized in neurodegenerative diseases to be a marker of neurodegeneration in cerebrospinal fluid. In Huntington's disease, it goes up in early symptomatic disease by about between 10 and 15% a year. So after 3 years, we would have expected those individuals to have gone up by about 40% in their CSF neurofilament from a lot of natural history data that we have. In the subjects who were treated, and actually at the low dose did have evidence of clinical improvement, but it was much less. It was the high dose that had the very clear effect. But these subjects had neurofilament below the original baseline before they started the study. So it was below about 30 to 35% of what we would expect. And so their neurofilament after 3 years was below what they started with. Now, that was bearing in mind they had neurosurgical procedure in the first year, which caused the neurofilament to go up because it was disrupting, it was putting catheters into the brain. If you have a boxing match, your neurofilament goes up 1,000%, and then it takes about a year to go back down to normal. So if there's any boxers, just be aware. You're actually causing some brain damage. So this went up, came back down after the first year, and now it's below their original baseline. And the only way to explain this is that you're saving neural function. So this is an objective biomarker. So the safety summary, the gene therapy, it's a single shot, one and done, has remained generally well tolerated. We did have some drug-related SAEs. They were within the first weeks post-treatment. It's a high dose of virus going deep into the brain. They all fully resolved with steroids or symptomatic treatment, and they were all inflammatory related. And we've had no new drug-related SAEs since December 2022. So in summary, the high-dose AMT met its primary and key secondary endpoints at 3 years. We'll have 48-month data in August, and a statistically significant 75% slowing of key clinical measures that have clinical meaningfulness for patients. And the CSF neurofilament was below baseline. And we did a sensitivity analysis, which was consistent with the primary stats analysis. And it's generally well tolerated. And actually, when patients, the patients who volunteered for this was that we didn't know whether it was going to be very dangerous. It was a single shot. It was one and done. And those were the people that were the real heroes. The people who came along were willing to have that brain surgery for something that we didn't know whether it was going to work and is irreversible. So I'd like to thank the clinical trial sites, and I'd like to thank you for listening.
Absolutely fabulous. Thank you very much. A brilliant study and fabulous results. So, yeah, open the floor to questions. We've got a couple of minutes of questions before we go to the break. There's one just here.
Happy to talk about regulators. It's a very topical subject at the moment.
That was a really great talk, and it's really fascinating. I was just wondering, obviously you've probably considered it, but could you treat the patients before they get symptoms?
Oh, well, that's a subject close to my heart.
Watch this space. Yeah, watch this space. No, yeah.
Watch this space.
Yeah.
I have been, that's exactly what I want to do. So that is my main goal currently for the next 10 years of my life, in that I want to do a prevention study in people who are stage 0 or 1, who have no symptoms but are clinically well. Because I think, and knowing enough about the biology and science behind Huntington's disease, I think will stop and prevent neurodegeneration occurring. So the gene therapy is not very practical for that, because I have a cohort of stage 0 and 1 patients, and there are about 65 people. It costs about $1 million or pounds per treatment. That's not what it's going to be. That's currently just to do it. So it's just not practical to do that. But I'm working on another approach, similar, same target, with an siRNA against exon 1 that will do the same, and that's exactly what I want to do. I think now we're at that stage we should be moving forward to a future prevention trial. So thank you for the question.
Didn't plant it. Yeah, just one final question I have, Sarah. What's the next steps here? Is it approval, regulatory? Where
yeah, it's a very good question. Yeah. So we had been having a lot of discussions, and I know someone mentioned Peter Marks. We had a lot of discussions with Peter Marks and Nicole Verdun at the biologics side of the FDA, who were immensely supportive. They approved the external comparator group. They gave us lots of ideas, and it had RMAT designation for fast track. As many of you know, there's been a change of government in the US, and that has resulted in the FDA on the biologics side and the gene therapy. Nicole Verdun left, Peter Marks, I think, was fired, and so there was a massive change on the CBER side, and the new management with Vinay Prasad and so on, although he's just gone as well, is a little bit up in the air. But it is ongoing, but it isn't so straightforward with the FDA, but we hope it'll move forward. However, and this has made me very proud to be British, the MHRA, we have been discussing with them, and they have invited us to put forward a marketing application authorization. And just to think, I don't know if anyone in the MHRA is here, but I just wanted to say that I was super impressed, because when we went to the MHRA, 3 of the clinical assessors, and it was a very formal process, came to Queen Square to the hospital because they wanted to see how all the rating scales were executed, how they were seen in patients, how they were measured, and what a matched control would be like. So they met a number of my patients, we did all the assessments, and so they went away 3 hours later knowing exactly what all those measures were. And I've never had a regulator do that before, so hats off to them. So that's hopefully the MHRA.